All right, let's take a look at the history of the DNA molecule. We will explain how the genetic code is contained within the DNA and investigate the explanation of the DNA structure. DNA stands for deoxyribonucleic acid. DNA is present in the nucleus of all eukaryotic cells. DNA controls the chemical changes which take place in the cells. The DNA tells the cells what they're responsible for producing, whether they're going to make muscle cells, blood cells, nerve cells, skin cells, etc. The DNA is also responsible for determining the type of organism that is going to be produced, whether it's a buttercup, a giraffe, herring, human. The DNA molecule, DNA, remember, is one of our macromolecules. It's an example of a nucleic acid. The subunit or the monomer for a nucleic acid was a nucleotide. The nucleotide, remember, has 3 parts: the phosphate group, the 5-carbon sugar, and a nitrogen base. When we look at the DNA nucleotide, again, we know we have that phosphate group. The 5-carbon sugar is going to be deoxyribose for DNA molecule. And then our nitrogen base. This diagram is showing us adenine, but remember it could also be thymine, guanine, or cytosine in a DNA structure. DNA has a double helix. If you imagine this is a ladder, this is where you would put your hands on the ladder, and then going across, this is where you would put your feet going up and down the ladder. And this double helix, they've taken the ladder and it's twisted, and that's the double helix structure that you get. Where you would put your hands on the ladder, this is where you'd find your phosphate sugar on both sides. Phosphate, sugar, phosphate, sugar. And then coming off the sugar molecules are the nitrogen bases. Thymine goes with adenine, cytosine pairs with guanine, adenine again with thymine, we have thymine again with adenine. Those are the nitrogen bases: adenine, thymine, guanine, cytosine. Our nitrogen bases are grouped into 2 categories, and this is something that is done in organic chemistry. We have the purines and the pyrimidines. The purines are going to represent adenine and guanine, and you'll notice that the purines, they have 2 parts to their structure. The pyrimidines only have 1 part to their structure, and that's going to be thymine or cytosine. Again, looking at our DNA structure, imagine this is a ladder. It's laying on its side, leaning up against the side of the house. The backbone, again, you've got your sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's like where you would put your hands on the ladder. And then coming off the sugar is where we would find our nitrogen bases. Here's adenine. On the other side, we've got thymine. We've got thymine here. On the other side, adenine. Guanine pairing with cytosine. Cytosine pairing with guanine. They're bonded together by hydrogen bonds. Those red dotted lines in between those nitrogen bases are representing the hydrogen bond that's allowing for the nitrogen bases to combine together. Discovering DNA was a team effort. There were several scientists and different experiments that led to the DNA and knowing that it carries our genetic information. Beginning with Frederick Griffith. He asked, how is it possible that bacteria can make people sick? And through his experiment, he was able to find that it was through a process known as transformation. His experiment involved looking at bacteria being injected into mice. And what he had was a form of bacteria, but he had two different strains of it. He had a rough strain and then a smooth strain. And when he injected the mice with the rough strain, the mouse was able to survive. It lived. But when he injected the mouse with the smooth strain, he noticed that the mouse died. He then took that smooth strain and heat killed the smooth strain and injected the mouse, and again the mouse was able to survive. So then the idea was, let's take rough strain where mice can survive and heat killed smooth strain, again where mice survived, mix them together and inject the mice, but in this case the mouse died. And this is what led to the discovery of transformation, since there was a mixture of two different strands of bacteria, both individually allowing the mouse to live, but when they were mixed together, the mouse died. So this led to the idea that transformation was occurring, which means that one strand of bacteria is passing on genetic information to the other strand. That smooth strand originally killed the mouse. So something, even though they killed that smooth strand bacteria, something in the genetics had to be passed on to the rough strand in order for that mouse to die. Okay, Oswald Avery continued the study of Griffin's work. He wanted to know which molecule was important for the transformation. In his experiment, he was able to determine that it was the DNA that stores and transmits genetic information from one generation to the next. He took a sample of that lethal bacteria that was causing the mice to die. He then was able to go through, and separate out the individual components, the lipids, polysaccharides, proteins, and nucleic acids. He then removed one macromolecule at a time to see what would happen. He removed the lipids, did a culture. The culture showed it to be harmless. However, the mixture containing polysaccharides, proteins, and nucleic acids still showed the culture with a lethal substance. He then separated out the polysaccharides, did a culture of the polysaccharides, and it was harmless. But again, the mixture remaining with proteins and nucleic acids, which still show a culture that was lethal. He then removed the proteins from the sample, looked at a culture, it was harmless, but then the nucleic acids were left alone. That culture showed that it was lethal. Further experiments, he was actually able to separate out the RNA from the DNA and did cultures of those individually. RNA showed to be harmless, but the DNA continued to show a lethal substance. Therefore, that is helping scientists know that DNA is again responsible for the transmitting of genetic and storing of information, passing it from one generation to the next. Hershey and Chase continued with the discovery of proving that it was the DNA that was responsible for the genetics. They took a bacteriophage, which is kind of a virus that can infect a bacteria. And a bacteriophage has this DNA core, which is going to contain phosphorus, remember your phosphate group, and then it has a protein covering on the outside that contains sulfur. Let's look at the experiment. Hershey and Chase experiment, they took the bacteriophage, and in one case they were looking at the protein. On this side they're looking at the protein, and they allowed that bacteriophage to attach to the bacteria, and then they noticed that there was no sulfur detected in the cells at the end. Since there's no sulfur, they knew that the protein was not responsible for passing on genetic information. Then they looked at a bacteriophage. They were looking at the DNA core, which again is going to contain that phosphorus, allowed it to attach to the bacteria, and the bacteria became infected. They were able to detect phosphorus inside the cells, indicating that it's the DNA that's responsible for passing on that genetic information. Erwin-Chargaff, known as Chargaff's rule, tells us how the DNA nitrogen base pairs are going to pair together. Remember, adenine must go with thymine. A and T go together, apples in the tree. Guanine goes with cytosine, it pairs with cytosine. G and C, car in the garage. The amounts of the DNA molecule that will be present are going to be about the same. When we look at the base pairing for diagrams, this is showing us guanine bonding with cytosine. Again, those dotted lines connecting them, those are our hydrogen bonds. And then here's a thymine bonded together with adenine, again by hydrogen bonds. For Chargaff's rule, again, adenine bonds with thymine. Guanine is going to bond with cytosine. Again, imagine this, your DNA structure as a ladder. On the side, you've got your phosphate sugar, and then your nitrogen base comes off of that sugar, and they're going to bond, again, adenine with thymine. Don't try to memorize that you have adenine followed by cytosine, followed by guanine, followed by thymine. That changes. There isn't a set pattern for that. That's going to indicate more about our genetics, and we'll get into that in our next unit. What you guys need to know is that adenine is always going to bond with thymine, and cytosine is always going to bond with guanine. Rosalind Franklin used X-rays to determine that the DNA was a spiral shape, and then later Watson and Crick came along and said that they made the model for the DNA, the double helix model, which actually won a Nobel Prize. The last thing I'd like you to do is you're going to draw the complementary strands for the DNA nitrogen bases that I have given. I'm going to do the first one for you, and then you will do the last three. And this is a common question that you will see. You're pretty much guaranteed to see this on your EOC, where they'll say draw the complementary strand for the DNA, and they would give you nitrogen bases, and you would say cytosine is going to pair with guanine, adenine pairs with thymine, guanine pairs with cytosine, guanine pairs with cytosine, cytosine pairs with guanine, cytosine pairs with guanine, thymine is going to pair with adenine, adenine's going to pair with thymine, cytosine's going to pair with guanine. Now you guys do the remaining three, and we'll go over those in class tomorrow.